Oh, this is a very good diagram. Just focus on this, will solve all the questions. Let us see, we are talking of the capillary. Okay, we are talking of the capillary. In capillary, normally the in any capillary anywhere, what should be the osmotic value? 280, good, 280, 300. Okay, but now why is author indicating 700 here? Because Vasorecta, we are going down. Okay, so this is 3. Okay, now along with that, along with the blood, okay, from this side, along with that, the author is bringing what hormone here? Vasopressin. Vasopressin will get out of Vasorecta, it will get on the interstitial fluid, it will act on the tubule that is going down, okay, and there it encounters a receptor. Okay, can you see this receptor? Where is it sitting? It is in the plasma membrane. What do you call that receptor as? Vasopressin receptor. Now this is again typical peptide interacting with this receptor, 7 transmembrane. Hello. Okay, it will interact with the, so this vasopressin is the ligand. After, as a result of that, this is G protein coupled. It will give rise to stimulate adenyl cyclase. It will give rise to cyclic AMP. There is a signal, and finally the signal goes to the synaptic, not synaptic vesicles. These are the vesicles within the cytoplasm. Okay, and sitting on those vesicles is the, what can you read there? Aquaporins. Now, under the influence of the hormone, these will be, there will be what? Trafficking will be excited, cytoskeleton system will be excited, and those vesicles will move, and finally where have they gone and settled? Apical or basal? Apical. Now suddenly for the water, okay, aquaporin is available. Okay, so through this aquaporin water will get in. Okay, now this is, I will say, inducible aquaporin. Who induced it? Vasopressin. Okay, at the this is on the apical side. On the basal side, there is another aquaporin which is constitutional. So it's always there. So the water will come in, and the water will get out, and the water will get into the blood. Have you followed the entire sequence of events which are triggered by what? Which are triggered by vasopressin. Look at this beautiful image author has taken. Look at the artistic composition behind this figure. Okay, let us see. This is the apical side, this is the luminal side. Okay, same thing, what we did in the earlier slide. Okay, the blood is here, the Vasorecta somewhere is here, and what will come out? The vasopressin will come out. Okay, and can you see that green receptor going 7 times up and down? Okay, it is, we have some more detail. What is it called as? Can you tell me what is the meaning of AVPR2? Can somebody tell me? Arginine vasopressin type 2. Arginine vasopressin type 2. There is V1, there is V2. This is what type? This is V2. Okay. Above that, can you see that tiny, what has author written there? Aquaporin 3. You know, in the earlier figure we had seen aquaporin 1, 2, 3, 4, 7. We had seen, out of that the author specifies what kind of aquaporin is constitutionally sitting on the basal side of the membrane. 3. It's there. Okay. All right. So then arginine vasopressin combines with the receptor, G protein coupled adenyl cyclase, cyclic AMP acting on the vesicle, then microtubules and the actin filament, it will move there, and then aquaporin 2, again aquaporin 2 will be introduced here. This is inducible. Okay, and this will allow the water to get in. And supposing you take water. Okay, as a result of that, now let us go the other way around. We have taken in water, so arginine, so those cells of excretion vasopressin are now inhibited. As a result of that, arginine vasopressin level in the blood has gone down. As a result of that, you don't get that stimulation anymore. Okay, shortly within 10 minutes, those vesicles will be taken in. Okay, and no more aquaporins are available for transport of water from the lumen to the inside. Are you with me? So for this system to operate, they will continuously need signaling from arginine and vasopressin. Okay, are you with me? Because there is no point in keeping the aquaporin there. Okay, once the osmolarity has been restored. Make sense? Going back to the very important point. what are the factors that are responsible for building up the high osmolarity in the medulla, and we have given a lot of credit to the thick segment, thick ascending segment, and we said in that in the wall there are a lot of transporters, and because of which sodium, we keep on talking about sodium. Okay. Author tells us that the contribution of sodium and along with that other ions is about 60%, not 100%. What is our target? I want to understand what are the factors which are responsible for building the osmolarity to 1200. My candidate number 1 is sodium. Does it give me 1200? No, it does not give me. It does not give me 1200. It gives me about 60% of that. Then which is the other important character I am missing? The other important character is, strangely enough, what is it? Urea. Urea, which we normally consider as a useless molecule which needs to be thrown. Okay? A time has come for us to slightly change, okay, our concept about that molecule. Okay? Within limits, urea is a useful molecule that contributes to building up the osmolarity, high osmolarity in the medulla. Did you get the message? And it is so important that it contributes almost 40% of the osmolarity in the medulla is because of urea. It is something like this. The kidney has thought into inverted commas. Okay? The kidney has thought that I am here to separate the urea, good. Can't I put it to some use in a restricted way? What can, well, urea may be otherwise an excretory product and needs to be thrown, but it's a solute, and that solute is in a sense useful, because if I want to increase the osmolarity in a particular area, that molecule is very helpful. Okay? Forget about this, I will tell you a story. Forget about all this. You know shark? Shark. The shark lives in the, it's a seawater animal, seawater fish, it's a fish. Okay? Shark is a fish, cartilaginous fish, chondrichthyx you can call it. Okay? Now, osmolarity of seawater is very high, we all know it's salt everywhere. Osmolarity was 1000 seawater. Okay? It is more than 3 times stronger than the osmolarity of your blood. Your blood is 300, it is 1000. Okay? So every most of the fish, other fish, bony fish are continuously being subjected to dehydration because their osmolarity was 300-400 fish, and outside is 1000. They are continuously losing water. You are in water, but you are dehydrated. Sea water. Okay? Water everywhere, not a drop to drink. So, but in shark, the osmolarity is 1025, 1050, 1075. So, the osmolarity of shark blood is even stronger than that of seawater. So, that guy has no problem. Okay? He can survive very well. Okay? Now, you can ask how the animal has been able to maintain such a osmolarity, shark. The answer is urea. Urea. The blood urea concentration in the blood of a shark is 100 times more than your or my blood. Then you can ask another question, how does that guy tolerate that urea? Okay, that is another question. We will talk about it in animal physiology too, if you are still there. Okay? But why am I making this point? I am making this point because I want you to appreciate the beauty of nature, okay, in trying to find out a solution if you can find any. Okay? So urea molecule actually it is what? It's a every cell generates ammonia, that ammonia is ammonia, you can excrete ammonia, or your liver can convert what? No, I made a mistake, I have corrected it. The joke is costly because I have lost my theme of thought. Such words always create a problem.
Your liver can convert.
Liver can convert. Ammonia into urea. Ammonia to urea. Okay. So, the nature has thought, why can't I use that molecule, okay, for taking my osmolarity so high, okay, so that I am not really worried about the osmolarity of seawater. Okay. Having conveyed that point, I will come back to this point. So, what is urea? Urea is Let's see now. Okay, so the kidney handles the urea in such a particular way that that the kidney hangs on to urea for a while, okay, within the medulla of the kidney, okay, and which contributes to about 40% of the. So urea plus sodium all put together is about they work out to about 12, taking the enriching the osmolarity of medulla to about almost 1200 milliosmoles. How does that happen? Let us follow. First of all, look at the thick line. Look at the thick line. Look at the image. Look at the thick line. That is the part wherein the urea is locked and it can't go out. Are you with me? Is it dependent on aquaporins? No. Aquaporins is for water. This is for urea. Okay, but and its profile is not exactly like aquaporins. No, it's a different profile. So you this part of the nephron where you must remember as to where urea can get out of the nephron and where urea cannot get out of the nephron. Did you get the image there? From that thick line, urea cannot get out of the nephron. Are you okay so far? Okay, good. Now the ultrafiltration is happening. Now the author has given a image, given the number 4.5. Now what is this 4.5? Very simple. Or what is the osmolarity of blood? 280, 300, whatever. Out of that, the contribution of urea is 4.5. You got the language? Okay, lot of it is because of sodium, lot of calcium, very little because of potassium, because blood potassium is very low. All put together, I am not worried about it. I am only worried about the urea, and urea contributes to how much? 4.5. Are you good so far? Now, as you go down, it becomes 7. Okay, water is absorbed, other things are absorbed. Okay, 7. Hello, are you with me here? As you go down, it becomes 15. Why? Because does urea go? It also goes. Water goes, but urea doesn't go as much as water goes. Therefore, it becomes 15. It becomes 15. Okay. Now, just don't bother about these arrows. I'll come back. Then you go here. Okay. As you go here, okay, then the urea can't go out. Okay, and then as you go by here, you have about urea, which is about same as that was filtered, and then you go here and go here. All other things are being absorbed. Okay, by the time you come here, the concentration of urea has gone to about how much? 300 milliosmoles. Now, this part of the medullary collecting duct is equipped with urea transporter, something like aquaporin, but the different molecule. Urea transport, what will it do? It will allow the urea to come out. Now, are you in the cortex or in the deep in the medulla? You are deep in the medulla, you are right. So, deep in the medulla, the kidney and urea concentration is very high. So, the urea, because of the transport, it flows everywhere in the medulla, and these cells in the loop of Henle, they have got the capability of picking up that urea and secreting it in their lumen. So, you have a urea cycle. So, whereas some of the urea is being thrown out, some of the urea is being recirculated in which part of the kidney? That adds to about 40% strength of the medullary fluid, so that it can. It's a very interesting point that is covered by arginine and vasopressin, and I will talk about it in this very, very interesting slide. Please focus on this. Entire world knows that arginine and vasopressin comes, it acts on those cells, it brings about it induces the aquaporins will go on the apical membrane, water will be absorbed. What is less known about the property of arginine and vasopressin is that it also facilitates the reabsorption of urea, because if urea is not there, then you will lose that 40%. Hello, are you with me? Urea is contributing how much? 40%. If urea is not there, you will lose 40%. That is not a good idea. So, if you want to, so what is your problem? Your problem is you are a land animal, you are very often subjected to dehydration. Okay, and then you have to have very powerful mechanisms of reabsorbing water. And what is the mechanism of reabsorbing water? You have to make sure that your osmolarity of the interstitial fluid in the medulla is high. That is the only tool you have. If that osmolarity goes more and more and more, you can absorb more and more water and you have more efficient kidney, and the extreme example of that is the Australian mouse. Okay, it can why is it able to, why is the that mouse capable of generating such a strong urine? Because it is capable of generating interstitial fluid in the medulla whose osmolarity is 10,000, therefore it can absorb. Are you with me? So your capability of the kidney's capability of reabsorbing water of the fluid that is being thrown out of the body will depend on osmolarity of the, how much of osmolarity can you generate, that depends on that. Now, so here you have, so let us see, we are again looking at the same cell. What cell? Medullary cell. Of what? Collecting tubule. We are looking at what? We are looking at the basolateral membrane, and here is the receptor, same 7 transmembrane receptor, arginine and vasopressin, adenylyl cyclase, cyclic AMP, and this mechanism, and then finally you have vesicles. But these vesicles have again very large protein molecules. They are urea transporters. What are they? Urea transporters. Like aquaponics, they will also go and sit in the apical membrane, okay, and they will facilitate the transport of urea. Okay, so what are you really trying to do? You are trying to retain the urea into the medulla. When urea is retained, it will increase the osmolarity. Once you increase the osmolarity, it adds up to your power of reabsorbing water and eventually conserving water. Am I done there? Hello, are you good?
Sir,
Yeah.
since vasopressin can stimulate urea reabsorption, can under dehydration the osmolarity of medulla increase even beyond 1200?
No, it does not. That is the limit. Yeah. That is, we believe that the human kidney can go from 1200, 1300, 1400, but that is the range, it cannot go beyond that.
Okay. So during dehydration levels, it slightly fluctuates from.
These are individual variations. When you say 1200 to 1400, these are individual variations.
So it more or less same remains constant.
Yeah, it remains constant. Like any other thing, like any other feature in your physiology. Yeah, we are always talking about a range. Yeah.
And in the case where we have to make dilute urine, there is low vasopressin in the blood. So does urea transport also decrease in presence of low?
Absolutely. The amount of urea in the medulla is being governed by, okay, arginine and vasopressin governs 2 things: absorption of water, okay, and making sure that urea is retained. So when vasopressin goes down, both the functions will relatively go, because we don't need them now, because you have had water. Okay, and whereas okay, whenever there is a challenge on your homeostasis, okay, our physiology responds by, just now I spoke about 10 minutes back, our physiology responds by 2 ways. Number one is a physiological response, which was, that's what we seen. Another was what? Behavioral response. Okay, and how is behavioral response generated? Okay, well, the information from the information is processed in the brain, and then the brain triggers the thirst mechanism. Okay, the thirst mechanism will do lots of things. You know what? Very interesting. In our mouth, our saliva is continuously being secreted at a particular rate, okay, and as long as the rate is normal, okay, our mouth is not dry. But when our body starts dehydrating, the rate at which our saliva is being secreted in our mouth goes down. Just when the saliva is still there, but the rate at which it is coming, when this goes down, generates a feeling of dryness. Generates what? Dryness. My throat is dry. Okay, is it really dry? No, saliva is still there. Okay, but it's not coming at the same rate as it. And then the moment it's dry, okay, it will trigger what? Then what will you do? Start looking for water, okay, and ingest water. Recent studies show that osmoreceptor is also there in our throat, okay, and in esophagus, and in different parts of our alimentary canal. Osmoreceptors are there. We don't know much about it, okay, so don't ask me question, I don't know. You see, even there is loss of blood, accident, loss of blood, okay, there is dryness in mouth, okay. Is there a change in osmolarity? No, Okay, even the loss of blood can generate thirst mechanism. Let us see this, very interesting. The plasma osmolality. We are normal, we are somewhere here. Okay, we are, what is that? That is arginine vasopressin in the blood. Okay, as the osmolality goes here, what happens to the vasopressin? Can you see this? Vasopressin in the blood. Okay, so you are collecting vasopressin when you are fully hydrated. Now you are dehydrated, vasopressin goes here. And some here, what does this indicate? Light, dark. Okay, what does it indicate? The intensity of thirst is going up, and at the top you can see what hormone you are able to see the structure of what hormone there. Okay, and you can actually count that there are okay. See, this is a very rare case in human beings, where as a result of the damage or some other reason to the posterior pituitary, the patient is not capable of secreting sufficient amount of arginine vasopressin. What will happen? Can you guess? No, lots of urine, obviously correct. What is it? Lots of dilute urine. Okay. Now the patient has to, therefore he has no control, because he cannot, his kidney cannot absorb water. So the only thing the poor patient has to do is to keep on drinking water. Is okay, but keep on drinking water and formation of large amount of dilute urine. This condition is called as diabetes insipidus. Correct. What do you call it as? Correct. Diabetes insipidus, which diabetes refers to the problem with the urine. If it is sugar, it is mellitus. If it is water, it is insipidus. Okay. Now, what is we are grateful to the technology, because again biotechnology product has come up. What do you call it as? Vasopressin. This is a chemical name. Okay, this is like arginine vasopressin. Okay, and this can be used as for the treatment of the patients. Okay, just a couple of slides, I will complete my today's lecture. We have, so I will ask you a simple question, fill in the blank, tell me the name of a hormone that will help us to, no, tell me the names of two hormones which will sequentially help me to conserve sodium. Very good. Before aldosterone, what will come? Angiotensin. Very good. Okay. Now we will go to the other end. Other end is MOS. You have taken too much of salt. What is the other end? You have taken too much of salt. So how would the physiologist will, you remember something we had seen that graph in which you have increased your intake of salt 10 times. You remember, from 30 to 300. Okay, and so how does the body deal with excess of salt? The answer to that question is you take salt. Okay, osmolarity of blood goes up. As a result of that, the blood draws water from the rest of the body. Are you with me? As a result of that, the blood volume increases. That blood volume, increase in the blood volume is sensed. The blood goes back into the, follow the chain of events. Blood goes back into the right atria. Right atria now stretches more. This more blood is coming per beat. Okay, it realizes is my word. It realizes that there is per beat more blood is coming. Okay, it responds. Okay, to this kind of stimulus. It responds by generating hormone. And what is that hormone called as? Please read for me. I mentioned about it once in the past. What do you call it, please? Atrial natriuretic peptide. Is the term self-explanatory? Atrial, natri is sodium, okay, and diuresis refers to natriuretic refers to excretion. So this is a peptide hormone that is coming from the right atrium, okay, and of course it's a hormone, it gets into the blood, it acts on the kidney, okay, and what it does is, you remember we talked about ENAC. Do I make sense? What is ENAC? Epithelial sodium channel. Correct. Can you read? Can somebody please read for me the statement that is written in blue font? ANP inhibits ENAC located in the collecting duct. What was the function ascribed to ENAC? Sodium reabsorption. Okay. Now, what is ANP doing? Inhibiting, which means what will happen to the sodium? Not reabsorbed, it will be excreted. Okay, this is one of them. There are many, there are a lot of other functions all put together. So, this is your physiological response to make sure that when you have taken excess of sodium, okay, you need to get rid of it, and atyl natriuretic peptide accomplishes that function by inhibiting those systems which reabsorb sodium. Okay, I think I am at the end. There are a couple of slides I want you to read on your own. Just this tells us as to how the peptide is released. This gives us as to how the hormone works.